Just want to introduce the final panel. So we have a great array of practitioners here, and they're going to take a few moments to answer some questions that are kind of central to the webinar today, and then we're going to toss the doors open to the Q&A. So please do start writing your questions in the chat. And so the questions that each of the panelists are going to take a few moments to answer are: how do genetic considerations figure into the work that you do? What are the biggest data gaps or areas of uncertainty? How do you balance or incorporate uncertainty and risk in planning and action or no action? And what is your top research policy or other guidance need?
Should I go ahead and start, Andrea?
Please do, yes. Hi, Naomi.
Sure. Hi, everyone. I'm Naomi Fraga, and I'm the Director of Conservation Programs at California Botanic Garden down in Southern California, in Claremont. We were formerly called Rancho Santa Ana Botanic Garden, which you may be more familiar with, and changed our name this year right at the onset of the pandemic, so you may have missed the memo, the California Botanic Garden. These are really excellent questions, and in terms of how genetic considerations figure into the work that I do and my team does at California Botanic Garden, I am fortunate to work with a really diverse team and oversee a broad suite of work in plant conservation. And the work ranges from rare plant research, including monitoring, surveys, and genetic studies. But we also have a very extensive seed banking program. We have the largest seed bank dedicated to California native plants. And we also conduct a number of restoration projects and studies, from outplanting to seed-based restoration. Personally, I also conduct plant research. I conduct research in plant systematics. So considerations of cryptic variation, historical and contemporary gene flow across the landscape, and all of the genetic considerations presented across and today, and the research presented today really permeates my thinking as a person who actually conducts conservation on the ground. And especially with respect to taxonomic recognition of lineages, since quote unquote species or described taxa are the unit that we work with on the ground. I think a lot about what is recognized and or how diversity is partitioned and whether or not that actually informs us to do really quality conservation work. The diverse research presented here today, I think, is really central to how we might need to approach on the ground conservation of the flora. So I'm really glad that all this data, all this research was presented here today. But to sum that question up, it's really just, I think the very genetic considerations are a really significant component in guiding our protocols and studies we initiate and undertake. With respect to data gaps and or areas of uncertainty, I want to focus on sort of seed conservation and restoration with respect to that, because I often think about, I'd really like to see a lot more empirical studies that gather molecular data to track seed conservation and restoration projects and their process and protocols, similar to what we saw in Andrea's study, which I think that was really great to see sort of what are the consequences of these two different methods of propagation and whatnot. I am concerned about inadvertent and unintended human selection as a consequence of our methods, despite our best effort to limit those impacts. And I'd like to see more studies funded and conducted that will help us improve our methods and mitigate those issues. Some of our best protocols, for example, seed collection methods, often rely on somewhat dated models. So further, but also in addition to that, I'd like to see more long-term monitoring to track success and failures of these restoration projects over time, because I think we oftentimes don't have those data readily available. And so I think about, for instance, and I'd like to see this also with respect to seed conservation. So for instance, if we think about genetic diversity of an annual plant and seed storage, and if we had, I'd like to know if a study like this exists. If someone knows about it, please send me the link. But it's something I think about with respect to the research potential in our seed bank, where we can compare a seed collection in the seed bank, maybe collected 10 years ago, with modern standing diversity, and look at, so there are projects to look at this overall, but I'd really like to start testing our seed bank and to see how they might be changing over time, or sort of their longer term value as backup collections to hedge bets against extinction, especially for annual plants. And then in terms of balance or incorporating uncertainty and risk in our planning into action or no action, I think this is a really excellent question about how to mitigate risk. I think especially, I think a lot about this in terms of rare taxa, because I'm concerned that perceived needs often outpace the information that we need to address those needs. So I think a risk assessment of perceived needs should be considered strongly, but also just the fact that we need a lot more upfront data gathering, but I understand that there's oftentimes budget constraints and that's not always possible. But again, having those empirical studies, I think would help us better inform those projects rather than just working in a black box. I really liked Michelle Dudash's decision tree as a kind of tool to determine course of action for rare plant species. And yeah, those are my, oh wait, what is my top research policy or guidance need? I would really like to see region-wide guidelines developed for seed-based restoration, seed conservation, as it relates especially to seed increase in seed bulking.
Okay.
Go for it, Heath.
All right. So I'm Heath Bartosh, principal and senior botanist at a small natural resource consulting firm in the Bay Area called Nomad Ecology. And we, do a lot of work in the natural resource realm, helping utility companies with their projects, a lot of water, power, and gas type projects that involve restoration, and then some other restoration projects for land trusts and conservation agencies and things like that. So today I'm coming at you from sort of the restoration aspect on the ground and kind of focusing on planting pallets, where I'm sort of answering these questions from that we were given. And I think Stu in the last segment had some a couple good phrases that he brought up that I think also frame some of my answers here in terms of natural selection still happening, and that we want functioning ecosystems. And just in terms of thinking about it from places that we can relate to, I think of Kings County, for instance, and I would put that in the, yes, I would like functioning ecosystems in Kings County because it's largely agricultural and there isn't a whole lot of native habitat there. Whereas, natural selection, you think about Alpine County and you're at the very top of the Sierra there, natural selection is going to play a role in whatever restoration aspects that you do there. So, yeah, focusing on planting pallets, the first question being, how do genetic considerations figure into the work that you do? Basically, trying to avoid unintended hybridization as a result of restoration projects is probably the biggest thing that we would really like to avoid in terms of genetic considerations. So, it's important to recognize seed sources of restoration stock and the natural local distribution patterns of native populations of the same species that you're working with. So I think that's our biggest consideration. And I think about it, like I said, in terms of local geography. And here in the East Bay, we've got some geographic isolation that presents itself and has resulted in a lot of endemism and possible cryptic diversity, like we heard about earlier, at places like Mount Diablo and both land masses across the Carquinez Strait. Those things, these are geographic isolations that have been developed and resulted in lineages of plant species over the years. And, those things are important to recognize when you're selecting plant species for plant palettes. And, another thing to think about is that, or that I think about, is that there has been a lot of development over the past 150 years, and that sprawling concrete jungle that has been created, does that present additional geographic barriers to gene flow and pollination out there in natural systems? So just some things to consider in terms of genetics. Then the next question is, what are the biggest data gaps or areas of uncertainty? And I think I'll just hit on something that is more of a classical nature, in that, we lack recent distribution and data distribution information supported by herbarium collections, and especially of common species that are usually used in restoration applications. So, observation data is great, but we all know that old adage that observations without a specimen are just a rumor. So the thing is, we're always out there. If you're a collector, you're either collecting for your rare plants or some other target that you're doing research for. It's just as important to collect common things that may be in the restoration trade, to create a benchmark and maybe provide opportunities for other research in the future. And the other data gap that I wanted to mention is that just genetic information and studies on, sort of less sexy common species, just like the idea of collecting these more common species. But, some of the work Christina Scherenbeck mentioned that she was doing on yarrow, that's great information, and a plant that's used widely in the nursery trade and restoration applications. So, get some academic advisors to steer more students to these types of restoration conundrums involving hybridization and gene flow for common plants would be awesome. Then the next question, how do you balance or incorporate uncertainty and risk in planning and action/no action? And again, thinking about it from sort of the restoration and building a planting palette exercise, it's about being conservative, really. And just to provide two examples of trying to be conservative is that I'll provide you with two plants here, and they're really sort of framed in this idea of restoration projects recognizing what's within a county. And so for one, Deschamps-ia elongata in Solano County, it's only represented by one collection in the Vaca Mountains. And would that be appropriate to then put in a restoration planting palette for coastal grasslands on the western side of Solano County, as opposed to where it's been collected in the higher elevation northeastern portions of the county? And then second, Festuca rubra, Point Melati fescue, something that's widely used in restoration, golf courses and all over the place. Is it appropriate to plant on the western edge of the San Joaquin Valley in Contra Costa County, where it's really only known from areas like that are on the bay side by Point Richmond and the coastal hills on the bayshore that experience that fog incursion from the ocean? So, being conservative there would mean that you try and remove these species from a planting palette, change the species. But, sometimes these planting palettes are already blessed and seeds collected and the project is underway. So I think sometimes it's hard to actually make that change or affect the necessary change you need to maybe make a really solid local plant palette for a restoration project. But, something to be considering there. Be conservative if you have to, and be early in the process of reviewing these things before they get too far down the road. Then the last question, what does your top research, policy, or other guidance need? And I actually have three. One of them, which has come up a couple times in the past year or two, is that I think we need some guidelines or policies for contract growing of rare plants, because a lot of times the traditional means of permitting rare plant collections don't really happen in a CEQA context. So you'll get approval from a resource agency to, or you'll get approval to collect threatened and endangered plant species seeds through CEQA action and not necessarily through an incidental take permit. And so, you know, where's the guidance and the policy related to those situations where you don't have some real regulatory oversight? And then after the seed is amplified and the restoration project's done, what actually happens to those excess seeds following the restoration efforts? And, you know, at places like Hedgerow Farms, you know, do they go out on the market to the general public, or is there any agency oversight at that point? And those are the things that kind of concern me, are people, you know, buying up these rare plant seeds and doing what they want with them here and there. It's maybe something I wake up at night thinking about. Then, you know, again, more efforts to collect voucher specimens statewide. And I think about San Diego Plant Atlas. I don't know if you guys have heard about this thing, but look it up on the internet. It was a great way that San Diego County organized, broke the county up into smaller quads, and people were assigned each quad to go and collect plant species that they didn't have information for, or do just general collecting in those quads. And what the result was a great deal of information on local distribution of all kinds of plant species, rare, threatened, endangered, and common, and weeds. So, you know, I think that's something that maybe CNPS as a whole hasn't really beat the drum about. And, you know, I think more counties would be into trying to do something like that. And then lastly, I think, you know, is there something that could be like restoration guidance counselors, so for, you know, representing certain localities or, you know, regions that, you know, could advise on, hey, you know, what do you think about these species in the planting palette? And so just having some sort of framework set up for, like, is there a place where people know where to go to get guidance on some of these plant species and whether or not they should be used in certain restoration projects in certain areas. So thanks.
I'm going to jump in because that was just so perfect segue for me. I'm Diana Benner with the Watershed Nursery. We are based in Point Richmond, and we grow container material for restoration projects. We started back in 2001, and very early in our time, there was a restoration nursery conference up in Davis, and I got to meet John Anderson and hear Kevin Rice speak. And it's my life has been really immersed in Phytophthora stuff recently, so I'm super excited to be here today and getting back to my first true passion of genetics and plants. And I'm very honored to be here and grateful to all of you for the research that you're doing, because it helps guide everything we do. And we are hired to go out and collect watershed specific material for projects. And so we are having to make the decision for projects of what to collect, where, and when. And we get the whole gamut of requests. We have some clients who are pretty well, who are savvy to these issues and are specifying really clearly what they want us to be, where they want us to be collecting, and other ones who don't really care at all. When we started the nursery, we were modeling ourselves on the Golden Gate National Recreation Area nurseries. So the collecting guidelines that they had developed in regards to the number of plants and the percentage of seed from a plant or a population, or what we were using in our protocols. Also having, and at that point in time, the GGNRA was pretty darn site specific in watersheds, and a lot of our projects were happening in more of an urban context, where, as Heath was mentioning, you know, we've got these cement infrastructure blocking flow between watersheds, and somewhat concerned about isolating populations as well. So trying to preserve local genetics, but also not amplify the isolation that was being created by what we've built here. Reading Malcolm Margellons and, you know, herds of hundreds of thousands of deer and animals moving, just imagining there was more gene flow historically than now. And as based on input discussions with Betty Young of how do you know, like which ones are maybe would be experiencing outbreeding depression or inbreeding depression would be really nice to know. So first research area of more information is just pollination, dispersal, and breeding system of plants. We are, as Keith mentioned, often on these projects not working with very rare species. So, and, you know, it might be things that scientists, researchers are walking past because they're not as sexy or something. But for us in our work, it would be helpful to know when we do kind of have to make the decision of we can't find the ideal population or what even define what is the ideal population for this project. Another area that was brought up today, and I was really happy to hear Christina Scheinenbeck is saying her name right, for the Platanus paper, because hybrids have been on our radar since the beginning, and Platanus in particular, and that's another area of research that I just feel nervous now when I go out. You know, for instance, if I'm getting rubus or sinus, it's like, is this true rubus? I don't know. I don't know how much to worry and what kind of breeding systems and polyploidy and all of that. So that's an area that would be great to have some more guidance around. Risks. Yeah, we have, so I mean, we have, like I said, clients who contact us who are not giving any specification to us at all. And so sometimes there's a risk of how, you know, where they're going to get the material if I don't have this specific area, seed from the specific area where their project is. So balancing that decision of, is it better to give them something that's from the next county over versus them going out and finding the material from someone who doesn't have a source at all, or who's going to sell them a cultivar, or who doesn't know where it came from. Or, so along those lines takes me to the top research thing, which is basically what he said. Like if we had what did you say, a restoration therapist local, but some, you know, professional panel or guidance that we could refer clients to, because they'll listen to us to a certain extent, but having more input from an official panel in regards to the species palettes that are put together, and also help educate. You know, this is a group of people who know about this, but we're dealing with folks who are not, you know, educated in all of the genetic complexities. I mean, when I took Bruce Baldwin's Jepson class last summer, my mind was blown by the sort of genetic uniqueness of populations of the same species, not that geographically distant. So anyway, very exciting, and having some more resources, educational for the layperson, and as he said earlier, input on species palette development for environmental consultants locally would be great. Thank you.
Great, thanks, Holly.
Yes. Can you see me? I'm only seeing Andrea.
I can see and hear you.
Okay, terrific. Well, thank you very much for having me on this panel. My name is Holly Forbes, and I'm curator at the University of California Botanical Garden at Berkeley. And like Naomi, although we have a much smaller staff and program, we aspire to the stellar reputation of the California Botanic Garden and Naomi's team. We are participating members of the Center for Plant Conservation. And if any of you are not familiar with their more recent public-facing information, such as the Rare Plant Academy, I'm putting that in the chat to everybody to become familiar with, because these are working guidelines, best management practices for a lot of our conservation questions. And at the UC Botanical Garden, we are working on a number of rare taxa, some of them down to literally one individual left, naturally occurring, some a little bit more, like 3. And then there are some populations that are sizable, but limited to one site. And so we're looking at some rather extreme basket cases, and genetic considerations are really important to us in this regard. But for the most part, we do not have information on the genetic diversity of the rare taxa with which we are working. We would like to have that. Sometimes we have a little bit, but the techniques that are now available, such as RADseq, that are allowing us to really delve deeper into individuals within populations, which, you know, most of the time what we're working with, there is just one population. So there's no between population consideration to be concerned about, unfortunately. But it would be helpful for us to know if keeping everything along maternal lines was as critically important as it should be or could be without this information. So without that information, we have to treat all of these populations as if each individual counts. And that's more work for everybody, but it also allows some of the research that has been talked about today or proposed in terms of studying older components of our seed banks with current seed health or seed production. We did have one study from Dr. Sarah Swope at Mills College, where she was able to take seeds out of our seed bank from well over 10 years ago, and then to compare the genetic diversity of that sample to what is being produced by the population today, and also to try to tie that to how the climate has changed in its native site. So there are a few of these projects going on, and we're certainly looking forward to more of them. We did hear about Dr. Chris Walters at the National Laboratory for Genetic Resources Preservation in Fort Collins, embarking on an IMLS-funded program with the Center for Plant Conservation to really ascertain the viability of seeds that have been in storage for more than 15 years compared to the viability of a fresh seed set. So we're looking forward to the results of that study in the next 2 years, which will help inform how we build our seed banks to preserve these rare species. In terms of our biggest data gaps or areas of uncertainty, like I mentioned, the genetic characteristics of some of these populations, we really just don't have that information. But really, for those of us who are trying to create new populations, learning what the microclimate tolerances are for these rare taxa is so important and so time consuming to learn. Because when you just have one reference site to base your decisions on to create new populations, you may really be limiting what the species can tolerate. And then, you know, kind of shooting yourself in the foot in terms of like, oh, well, I could have put it there. But you don't know that without a lot of experimentation. And we also need to spend a fair amount of time for all of these taxa with which we're working to determine their best germination needs. What practices can we employ so that we get the maximum out of our limited wild collected seed lots and to maintain the genetic diversity. Like Naomi, we work with quite a few annuals, and one of which is the large flowered fiddleneck, Amsdekia grandiflora. And we've had really great success working with Volmar Natural Lands Consulting to create new populations based on one reference site, or maybe 2, to determine where to locate it. But when you're starting with a very limited seed lot size, you don't want to have multiple generations in a nursery setting where you're actually growing things that prefer the nursery setting versus what might be happening out in the field. And so we limit ourselves to no more than 2 generations out if we can at all avoid any further, so that we're trying to keep that say genetic drift that might happen in a nursery environment to the most minimum we can. And when we use the seeds from these materials produced both in the field and in the nursery, we do not just go for the juiciest, biggest looking seeds. We go for all the seeds, because even if it's a really small seed, it might still just germinate fine. Might need a little help, but we don't know if there's some genetic diversity that caused it to be a smaller seed, but still is a component of the whole genetic makeup. So we're trying to keep those things in mind. And sometimes we're working with congeners so that we can learn how to grow something well before we risk the very limited rare seeds. We've just been doing that recently with a polygonum. How do we balance risk or incorporate uncertainty? Well, our first consideration is something that Andrea said this morning. It's like, first, do no harm. Sometimes it's hard to know whether that's going to happen or not, but we try to do our best to prevent extinction, right? So doing no harm. Well, well-intended conservation guidance in the past has limited some of our ability to collect seeds from these very small, rare populations. And it was the best of intentions, but that meant that we had a very limited seed bank with which to work when that population was badly damaged by others, inadvertently, but it happened. So instead of having, at least 50 maternal lines, we were down to 20 because the population was largely destroyed. So we do have some studies that show that we can collect seeds from these rare populations without unduly damaging their ability to reproduce on site, and more of those kinds of studies would be great to have. We are concerned about hybridization, and that makes finding new introduction sites for some of these rare taxa challenging, because a lot of them are just very happy to cross with their congeners, and finding a spot that's far enough away that the pollinators won't carry their pollen and create hybrids is sometimes a challenge. And in terms of our top research policy or guidance needs, well, the research includes, of course, the genetic considerations for rare taxa, the microclimate tolerances for establishing new sites for these taxa so that we're not down to just one. And one of the things that we've been really struggling with is grazing. There are a lot of these grassland species that are just having a tough time in our invaded non-native annual grasslands that used to not be such. And having grazing is essential for some of these taxa, because otherwise the non-native grasses just swamp them out. And the real challenge there for us is timing and intensity, and sometimes which animal. So that's a big need for a lot of taxa in California. In terms of policy, how do we best prepare for these changes in our climate? I mean, we have a tough time locating endangered species into a new site that's within the same climate envelope it occurs now, let alone trying to expand it into yet some other conditions. And most of the taxa we are working with now are short-lived. So it's not your ponderosa pine that will be perhaps okay, 50 years from now or 40 years from now when the climate has changed to better match what the seed source climate was. But these are short-lived, annuals, perennials. Do we give them the same sort of time frame of these climate models? And then disease issues are always a challenge. And as Diana mentioned about Phytophthora in our nurseries and best management practices and not introducing them into habitat are all big concerns and create a lot of work for everybody, but it's so important. So thank you very much.
Great, thanks. We have several questions to answer. The first is from Marley Montalvo. We need to always balance the risk of maladaption and hybridization effects with any potential benefits of carefully thought out assisted migration. There are plenty of models that help us understand which taxa or lineages are likely to harbor the most risk upon translocation. And I would like to see more discussion about risks and scale. The use of seed zones, such as those presented by Dan, provide us with guidance about the most logical way to move seeds in a way that is likely to minimize risk of maladaption over time. A random shotgun approach is much more risky for all kinds of reasons, not just for the focal species. Also, what climate model do we use for making predictions about future suitable habitat? Is averaging over many models realistic? And is there new work that points to which models are more likely to be the most correct?
This is Jim. I might make a comment about the climate models, if that's okay.
Sure.
So if you take the ensemble, then you basically are zeroing out changes in precipitation. So precipitation in some models is wetter and in others it's drier. And so if you use an ensemble model, you can still capture the increase in temperature, but you lose the range of future precip that a site might have. So I prefer to bracket and pick a wetter and a drier future, and then say, okay, this is within the range of 80%, let's say 80% of the GCMs that are out there for my site. And so but be very explicit about that and say, okay, here's what I think it's going to be. And then that lets you have something to work with. With regards to the other point, I mean, I think there's really a lot of urgency here, and that it's very well to talk about. We can try to advance as much as we can the understanding of the genetic relationships between multiple different populations. For things that are annuals and perennials, you actually have a better chance they might actually be able to adapt. But how fast will they adapt to changing conditions as opposed to long-lived species? But it's like conditions are going to continue to just kind of go along at the same way, and so you do nothing or you do something, monitor it, track what you're doing, because everything we do is an experiment and we don't know. And are you trying to save habitat or are you trying to save genetic diversity? And at some point it may be that you just have to try to catch the habitat because the diversity is going to be, the genotypes are going to be really that much harder to get. So if that's the case, then which ones would you take? Name me 15 species that are the most important. Let's go out and get that genetic data for those ones, and and go from there. Thank you.
Thanks, Jim. It was a little hard to understand a lot of what you were saying, so I don't know if you want to summarize in the chat some of your larger points. I'm going to move on to Kevin Rice's question. A lot of focus today has been on physical selective factors associated with climate change. How much do we really know about the role of biotic selective agents such as pathogens, competition, et cetera, in promoting local adaptation or affecting the phenotypic expression of genetic variation, that is evolutionary potential?
That's a doozy of a question there. And I'm not sure we really know. I think pathogens are probably the best understood and studied, because we've seen how pathogens can just rip through populations. And the competition, I think the point that Holly made earlier on about a lot of our grassland species just getting buried under annual grass and thatch. There's an environmental driver to that it's not been mentioned yet, which is the fertilizing effect of smog. So atmospheric nitrogen deposition is driving a lot of that excessive grass growth. So there's some really obvious competition things, little rosette forming annuals in the grassland can't make it when you have an inch of thatch growing over it. And as you just start adding up all the biotic interactions, it just gets so complicated so fast that, I'm not sure we'll ever know the answer, but maybe some general guidelines could be made to try to cover the bases. I forgot my video. Thank you.
Thanks, Stu. Does anybody else want to chime in before I move on to the next question? Okay,
Andrea, this is Tricia Maloney.
Hi.
I'm addressing Kevin's question. And so, I work with obviously insects and pathogens a lot. And, we know that there is adaptation to these biotic pressures because they've been interacting with, say, mountain pine beetle or even white pine blister rust, at least in the case of mountain pine beetle for, millennia and stuff. And so, it's a little song and dance that's going on out there. And unfortunately, a lot of the sort of genetic work, is, people are doing it, but it doesn't come out. And so a lot of my work right now is actually looking at sort of host defense chemistry and looking at the variation in certain populations of 5-needle white pines as, sort of an adaptation to a biotic agent like mountain pine beetle. And then there's even work with white pine blister rust where we know that, there is resistance in populations of sugar pine and in western white pine that are out there. And so those are the sort of, once again, biotic agents that have been sort of interacting with their hosts for a long time. And yes, there are adaptations to those. And yes, they should be considered, obviously, when you're planting out trees as well. And someone asked, I saw a comment about Phytophthora. So yeah, I'm just, like I said, just giving a little input into or Kevin's question.
Yeah, and the Phytophthora question was from Julie Coffey. Glad Phytophthora was brought up. Does anyone know of evidence of local resistance or adaptation to these pathogens? Might be a consideration for broadening seed sources. There's some thoughts I've heard here in Southern California that fungal pathogens in wet years may play a role in the spatial distribution of grassland versus coastal sage scrub.
So I know that Matteo Garvalato, who's been working on looking at resistance and whatnot in certain oaks and tan oaks, I believe, in Matteo's out of Berkeley. And then, one of the other things that comes up, and it was a paper done by the geneticist at Berkeley, Richard Dodd, maybe, where there's kind of two mechanisms for disease resistance. There's passive, or there's active resistance, which is actually like, the major gene for resistance to white pine blister rust and sugar pine. But then there's also this passive resistance where you have disease escape. And sometimes that can be just a asynchrony in phenology where trees are not leafed out or they're going into dormancy and their stomates are closed. And so, you have a dispersing spore and, it lands on a host tree, but because it's gone dormant, that spore doesn't enter the stomata. So, like I said, there are those sort of passive mechanisms out there, but and whether or not any of that's being looked at with a lot of the oaks and tan oaks in the Coast Range, that I don't know. And if someone wants to know more of the individual who asked that question about Phytophthora, I would get in touch with Matteo's lab and some of the others.
Yeah, and at the last Oak Symposium, Sudden Oak Death 7, I believe it was, there was some discussion about resistance and exploring resistance in a few species. And it seems like tan oak is 95% or more susceptible. The coast live oak, there's a pretty broad range in variation, and about 20% seem to be fairly susceptible 20% are fairly resistant, and then there's a pretty broad range in the middle. And this was, I think, some work by Stephen Swain at UCCE Marin, and he was looking at whether or not the phosphate treatment would be effective in stimulating resistance and prolonging the life of individual trees. I don't remember off the top of my head, but there is some similar looking at fatality in Quercus callagii, the California black oak, and then there are some other oaks in that clade that are fairly resistant. So the parvulas in particular. And I don't know if anybody else wants to talk about Phytophthora.
I wanted to talk about, I just remembered Carolyn Malmstrom's talk at the last conservation conference about viruses. I think it's a really good body of work that we could learn something from, because it gets, really
Hey, Stu, your audio was, oh, maybe it's my audio, was pretty broken up at the end there. If you want to type your comments into the chat, that might be better.
Sure.
Great. And just the last note about not Phytophthora, but Jessica, I don't know if you wanted to talk about some, and Patricia, you referenced this a little bit, some of the work that's been done in Europe about oaks and susceptibility to pathogens based on phenology. I haven't seen anything similar in the States though.
I think it's Richard Dodd, a paper that he did quite a few years ago. I think Katie, God, I'm blanking Katie's last name now. But I think it's Richard Dodd from Berkeley who worked with Matteo, looking at that sort of phenological disconnect with Phytophthora and oaks. If I find the citation, I'll send it to you and you can send it to the group.
This is Jessica. Can you hear me now?
Yes.
Awesome. I've been having internet problems all day. So yes, I've actually just been reading, I'm trying to write up the paper for the phenology data I presented earlier today. And there's a whole set of studies in France about how for Quercus rubra, that the timing of phenology is really critical in terms of avoiding getting hit by the powdery mildew, and that powdery mildew is it's a specific fungal pathogen to the tree species, and it only attacks a particular age class of leaf. And if the trees leaf out at the wrong time, then they are more susceptible to the powdery mildew. So there seems to be some combined selection on phenology by the pathogens to get the leaves large enough before the pathogen comes out. And there's Andre Kremer in his lab in France has done a lot of work on it, a bunch of different papers. I was involved with the Phytophthora resistance work in Matteo's lab, working with Katie Hayden, and we did a lot of inoculations of trees in the greenhouse, in the kind of grow chambers at Berkeley, and then we actually were able to take some of those trees and plant them out into nature and look to see if we had done a restoration project using our trees that had shown more resistance, if we'd had a more successful project, and our results showed that we pretty much would have. And I'll post a link to that paper in the chat in a couple seconds, assuming my internet holds up long enough.
Great, thank you. And Kevin Rice said, I used to try to ignore this, and I assume this means pathogenicity, until I watched 2 populations of Elamus glaucus go extinct in 2 years in a supposedly benign common garden due to rust infection. Arlie Montalvo has a couple more questions in the Q&A. One is, how is this information employed in Thorne's work? And I don't know if I can answer that because I don't know what this information is referring to. So I'll give Arlie a chance to ask another question there. And then the other one is a comment about working on a set of native plant profiles for plants commonly used in restoration in Southern California at lower elevations. There are still a lot of gaps. They can be a great tool for understanding particular taxa and groups of plants. Might we work on expanding a native plant profile program for important restoration species statewide? And I'll put the link to the profiles in the chat. I don't know if anybody has any comments on profiles for restoration species or how they see the best way to get this information out into the world.
I'm familiar with Arlie's plant profiles and have utilized them as a great source of information. Those particular profiles, as I recall, focus a lot on life history attributes, taxonomy, ecology, sort of a great summary of the diversity of knowledge you'd like to know about a particular species and undertaking plant propagation or, for a restoration project or implementing restoration project. Something we've also are working on in the garden, at the garden to complement, that would complement Arlie's plant profiles is also developing sort of alternate profiles that incorporate knowledge of propagation methods, including germination, germination information, seed and vegetative propagation, and all that sort of available information. I find that's a lot of the kind of questions we get at the garden is working with unusual species or species that aren't commonly grown in horticulture is getting success in propagating a diverse set of species.
Does anybody else want to address that? Or just where you get information on species that you haven't cultivated before, ways that you might address that? Okay, another question from Vince. Says, thanks. This is definitely not something to do casually right now, but it does seem inevitable that we need to consider it. I'm going to assume that Vince is talking about assisted migration and wanted folks to speak generally about one of the themes that I've kind of heard today, and that's the difference in adaptation timeframes for different life forms. So annuals and short-lived perennials being able to have some greater adaptive capability to the rapid change in climate that we're seeing now versus some of the long-lived tree species in particular. Does anybody want to?
I'll comment on that. Just that I think that, we always have this sense of climate change losers, but there's climate change winners out there. And thinking about past history and reading some articles out there, I think one in particular by Axelrod back in the 80s talking about, the time period after the Little Ice Age from 5,000 to 8,000 years ago, there was a period of warming in California that was a degree Celsius or a little above. And that assisted some migration naturally of Mojavian species up the west side of the San Joaquin Valley. And so, that implies that there's some genetic code that still resides in some of these species that, can handle some climate change and some migration on their own. And, personally, I always think that, sometimes just because people can do something, should they really be doing it? Without getting all the information available there. And I think that, that warming trend is a good lesson in that, and it being not too long ago, and that genetic material may be still fresh in the code of a lot of these plant species. So while yes, we will lose some species to climate change, there's also going to be some winners out there. And so how do we decide, who's going to, avoid the end of their lineage by our helping hand, when maybe for species like Amsinkia grandiflora, they were doomed to extinction not long after they speciated.
I'd like to weigh in on something Heath talked about, is the legacy of past climate changes in terms of these little we need the refugia, which will be the coolest, moistest spots. But the really arid spots on the landscape often contain little populations of arid adapted species that are the foci for expansion. So, that legacy of the Mojave species that we find in the Mount Hamilton range is just kind of fascinating. Things slosh back and forth across the landscape. You leave little bits behind. So.
Okay, and Brent, I see you want to say something, so feel free to unmute yourself.
Yes, while we're on this topic of assisted migration, there have been some comments also in the question and answer text. I do agree with what several have said, including Heath, that we need to be careful. In my opinion, it's not out of the question, absolutely, but it should be kind of a last resort once we really, it's our only hope, basically. What the point that Heath was making is that, and to amplify, there's not only genetic variation stored within these lineages, but there's a lot of plasticity, and there's a lot of microenvironments out there. So the annual climate, the annual macroclimate is not the only factor. It's whether the plant can find its microenvironment within that macro environment. And many of these plants can. So I think that without really good information, it's kind of like playing God to be moving these things around and turning the landscape into a botanical garden. I think what we ought to do is see if the lineages can migrate and undergo natural selection, like let's say ponderosa pine, and a good example, poster child. And then as a last resort, I think we'd want to do some assisted migration if it's just clear that that whole lineage is just not keeping up and not able to move, let's say change its habitat appropriately by natural means. So that's my feeling, is that I'm not completely against assisted migration, but I think that unless you know a heck of a lot about the biotic and physical environment, you shouldn't do it until you really are sure.
And Bruce, you wanted to say something?
Yeah, sorry, it took me a second to unmute. Yeah, there was something I wanted to say in my talk earlier and I didn't have enough time, but it's really relevant. I think that, we're really on the cusp of affordability of whole genome sequencing on a mass scale, and the accompanying development of much more powerful methods to analyze genomes in an evolutionary context, I think can really revolutionize our understanding of the evolutionary, ecological, and biogeographic history of California plants. And I really worry that large-scale restoration involving extensive redistribution of germplasm across the state could easily erase or hopelessly muddle this wild evolutionary signal, which Stu mentioned earlier. And I think if that signal remained available, it could potentially prove critical to responding most intelligently to climate change and other anthropogenic impacts on the flora. I don't think we should lose sight of the potential to really lose a major opportunity here and forever lose access to an extraordinary amount of knowledge about plant diversity in one of the most important global biodiversity hotspots. So I think we minimally need to have information, some kind of central depository of information about where human distributed plant germplasm is going and from what's original sources in order to have a prayer of trying to salvage any of the historical genetic signal from future field collections if we go down that road, which I hope we don't unless it's a last resort. And I also don't think we should forget the adaptive potential of plants also applies to standing genetic variation within existing populations. Already talked about that a little bit, but another factor is ecological release or competitive release. It's a real phenomenon. It's been demonstrated in Lasthenia by Nancy Emery and David Ackerley, where variation is not ordinarily seen among field phenotypes in a population is expressed when conditions are such that there's an avenue for those individuals to survive, such as in a greenhouse environment. And we've seen this growing out tar weeds from many different species over the years, all kinds of variation you don't see in the field. So I just think that we really do need to keep this precautionary principle in mind. And I'm just trying to pull us back to the first, to our morning session a little bit, some of those considerations, which, I think really do need to be kept in mind in all of this, because this has the potential to be a really major loss. It's not going to be a win-win if we go in and move germplasm on a mass scale. At best, it's a win-lose proposition. And I think we really need to try to, walk that tightrope and do this intelligently for this additional reason.
Thanks, Bruce. I wanted to ask another question from Kevin. As our climate becomes more variable, should we be looking for genetically more plastic genotypes?
I'll weigh in. I think probably yes. I don't know how well documented sort of the genetics of phenotypic plasticity have been worked out, but if there are individuals that can adjust phenotypically to a lot of different environmental conditions, if one would think that they would be selected for within populations. I'm not sure how we could do that a priori, but I think it's something that might get expressed when the big crunch comes. So.
Just to jump in quick, we may be seeing that happening with natural selection, since one of the outcomes of climate change, all models would say, is there's going to be a lot of variability between years, more than even normal. And, plants are famous for lots of phenotypic plasticity anyway, but I don't know that, we don't know enough about it. There's just a few groups that sort of study of genetics of plasticity have been looked in. So I don't think we're ready to do anything about it. And I think that nature may help us along there. I second what Bruce was saying, and I would prefer to let's see if these lineages can manage naturally and only do something drastic, like completely moving them to a new place, if it's really clear that's the only way to save them. The place to hang on to the genotypes is in botanic gardens. That's a very important role of botanic gardens, is to hold on, make sure that we have kind of a backup of the taxa. But as I mentioned earlier, personally, I'm not a big fan of turning natural areas into botanic gardens that we're kind of curating and moving things in and around and out.
Thanks, Brent. That we're kind of at the end of our time, and that gives me a really good segue. Today is Danzanita Day, as we call it at CNPS. That's the day that Dan Glusenkamp, I think coming back from climate change symposium, looked over after some Caltrans clearing next to a highway where some work was going to be done, and saw what was thought to be extinct in the wild, the Franciscan manzanita. So that's a really interesting kind of poster child for what we're talking about today. This is a plant that was not known to be alive in the wild. And through a lot of effort of a lot of folks, some of which are on the in the symposium today, that plant was dug up and moved. And there's been some work since then on both figuring out germination based on related congeners, and trying to resurrect some related species, or not related species, but get some seed from other plants to get some more genotypes into the wild. And so that's just a really interesting thought about, you know, some of the things that we're talking about, plants in the path of destruction. But something else that I kind of want to leave you with, that is kind of the goal of my work, is to not have somebody 50 or 100 years from now say, what were they thinking? So I think about the folks in the early 1900s who filled in the wetlands because swamps are useless. And I think about the CCC in the 1930s who did just tons and tons of tree planting, moving trees all over the place because trees are great. And now we're cutting them all down. We're restoring the wetlands. So I don't want to do anything that people are going to try and undo in another 50 or 100 years. So that's just kind of the thought that I want to leave you with today. I really appreciate everybody who has helped to make this possible. Once again, our speakers and the staff that helped to work on kind of the background logistics. This has been really great, and we're going to try and get the recordings posted up on our CNPS YouTube channel in about a week or so, and then we'll be coming out with some symposium proceedings that will maybe have some more information in it about some other resources that are available for guidance. So thanks once again, and I appreciate your time today.